let's do this one. Okay, so hello everyone. I just wanted to thank for the invitation. I really like when people can connect throughout Canada. I know that we are far away, but like it's very cool for me to present to new audience. I hope I can be clear enough with my research work and show you a little bit more of what I was doing. So thank you, Yasmina, for introducing my career. I just wanted to say that currently I'm in Vancouver, British Columbia. I'm still affiliated with the Genomes Science Center. So we are like leading institute in the sequencing of genomes, genome assembly, and especially we are like tackling non-model organism species. Just wanted to say that today I will be presenting my academic research that I did in forestry, but I'm really well connected with the startups in Vancouver. So if any one of you wants to get in touch with me, discuss about academia, research, or entrepreneurship, just send me a message. These are my contacts. I'm really happy to chat with you. And yeah, I'm just very open to any new connections. So today I'm going to talk about basically the forestry studies that I was doing at the Genome Science Center. So the first part, which is going to be focused on the conifers and on the trees part, is basically a study of the genomes of 4 spruces. And here, this is a picture of one of the most massive and like more important in the West Coast species of spruce. So this is Sitka spruce. And this is actually a picture that I did myself on the island of Haida Gwaii that I visited in September this year. The trees and the forestry is very important in Canada. It covers, of course, very important economical part, but it's also very important also for the indigenous people. So if you ever met this book, I really recommend it. So it's The Golden Spruce by John Vaillant, and it's the story of the sacred spruce that it's sacred to the people of Haida Gwaii. There is a lot of mythology about that. And the book tells this interesting story with, and basically this tree was felled in 1997. And it's the part of the ecology and mythology that connects to each other. So I really recommend it to read. So as part of my PhD thesis, I had the opportunity to work on the genomes of 4 spruce trees. So spruce are conifers. They are part of the gymnosperm family. And at the Genome Science Center, we sequence 4 genomes. We sequence 3 genomes for pure species. So we sequence white, Sitka, and Engelmann spruce genomes. And they are shown on the map. So the dots that are basically, those trees are sampled. And with the colors, you can see the distribution of where the species grow across Canada. So this is very important for the Canadian economy and for the forests. And with this project, we are basically trying to understand better the genomics of the trees. As you can see, they have very different features. As you can see, for example, the red dot, which is the Sitka spruce, it's a very high tree, reaching 50 meters of height, but it grows on the sea level, so it's in front of the ocean, and it has very different features than, for example, the Engelmann spruce that grows on the Canadian Rockies and grows on a climate that it's colder and more snowy. And on the other side, white spruce, that it's very widespread across Canada, and definitely this is a very interesting data set to work and to analyze. The 4th genome that I haven't mentioned yet is the interior British Columbian spruce, this black dot here. And it has been chosen because it's a very important tree for the BC forestry. It's very resilient, it's very resistant to pests, and there are several projects in BC that basically breed the interior spruce for lumber and for its like physical features. So we start with these 4 genomes, the sampling of the genomes. And the first hurdle that I want to introduce you is the fact that these conifers are actually, they are very difficult to sequence and to analyze on their genomes because they have a huge. So if you see like the content of DNA that we are often analyzing, it's mostly the human, which is in this part of the table. But if we go to conifers, it's basically going towards a very large genome size. And especially in conifers, goes from 20 to 30, even 40 gigabase pairs, which is, for example, in the case of spruces, 7 times the human genome size. And the genome, it's this gives a very big challenge for the assembly of the genome, and it's definitely a hurdle in that. Looking at this publication from 2013, that was one of the first publishing the Norway spruce, and they did a very good job in the analysis of that. It's possible to see that the genome size is big because of its repetitive content, and especially of the LTR type of repeats that in most of the cases cover even 60%. And in the case of spruce, we have more than 80% repetitive regions, which make it quite difficult to assemble and to analyze. For the genome assembly and annotation of the 4 spruces that I show you, the project started even several years before I came to the Genome Science Center. And it started with the sampling of tissues from these trees. So we have both diploid and haploid tissues. So the tree is diploid, but whenever you sample the haploid tissues, it's going to be easier for the genome assembly. So in conifers, that there are the megagametophyte tissues that are haploid and allow an easier assembly for example, for the scaffolding stage. So sampling the tissues, of course, there was a DNA extractions, and the genome has been sequenced, and in this case has been sequenced with short reads. So I'm going to present the genomes that has been assembled with Illumina short reads and 10x genomics. The part of the bioinformatics type of analysis comes with the genome assembly first stage, where we generate the string that represent the genome and the reference genome in this case. And the second part goes with the annotation of the assembled genome. So having a string of simple letters, we are looking for functional elements in the genome. And this is the reference annotations that we also submit to NCBI or dedicated databases as 3genes or Congeni. And the last stage of the analysis, it's a more comparative analysis that we did. So we are just comparing the genomes and the different type of features that we can extract from these genomes to look for example, local adaptation. I also posted the software, the link to the software that has been used to generate the genome assembly. So this is part of the GitHub repo at the BCGSC. And the group that I was doing my PhD into, it's the Bioinformatics Technology Lab. As for the genome annotation, I personally did a specialized pipeline for the annotation, since the genomes were quite challenging of these conifers. So this is most of my work, and the first years of my PhD I spent into improving the genome annotation pipeline of these conifers. Just to say that the genome annotation for one of these genomes, if just running the pipeline, lasts about 6 months. And this is basically not possible if you need to annotate 4 of them, and definitely if you want to do a different iteration of the annotation. So what I did, I implemented a strategy for the scaffold, I implemented a strategy for the filtering of the scaffolds. So from each of the genome, we have about 2 million pieces, and I selected the ones that have functional genes for the annotation. I trained myself the parameters for the annotation. So I trained the models for the HMMs in 3 software in the Maker pipeline, and I did 2 further iterations for an improved annotation. As you can see from this plot, annotation is definitely something that combines multiple data sets in order to get a good quality of annotation at the end. So I collected several RNA-seq samples and full-length cDNA, which I used as a supporting evidence. And I utilized all this data in order to create a high quality of annotation, since it's also very hard, given the repetitive genome, to find the genes inside, because most of the sequences, they are basically repeats. And we want instead to find proper exons and gene structures in the genome. This is a little bit of a summary of what is the results of the genome assembly. So on the upper table, you can see that the NG50, which is the way we classify the contiguity, or like how well are the pieces connected to each other. They are like basically in the same ballpark range. So we have mostly in the kilobase pairs of NG50, which is still at the draft stage because it's with short reads. But we managed to have a good reconstruction of the genome size, which is something that we expected from these genomes that we already know from previous studies. The completeness, so the Busco complete, it's another metric that basically checks for genes that are expected to be present in the genome based on homology. And we found them to be present in about 50%. And of course, we have the genome of Sitka spruce, which is a little bit less contiguous, and also the completeness is lower, but I think we managed to have like the same level of similarity in the contiguity and the completeness overall. At the annotation stage, I obtained about 28,000 genes, which is comparable within the different genomes. And also the median exon and intron length were quite comparable, I would say. Here, this is also another statistic for the completeness. And the completeness of the Busco for the annotated genes is a little bit lower, also because we didn't have the same RNA-seq evidence to use for all the genomes. But overall, we had a data set that we were able to compare to each other and to perform a comparative genomics. The comparative analysis we focus for this work was mostly for the study of local adaptation. Each of these genomes, they come from a different geographic region, they have different features. And I focus on the study of gene families and a positive selection on single copy genes. So I was looking of what it's actually different between my 4 genotypes or taxons. And I annotated several genes that are like significantly rich in each of the genotypes. So we have, for example, the transcriptional factors, heat shock proteins, other genes related to resistance to pests, and so on and so forth. So those are all already being reported in the literature. And this is like a first study at the genomic level that looks for local adaptation between different species, of course, but we have sequence 4 genotypes that we would like to compare in this case. And the same goes for the positive selection here for the genes that I also listed as transcription factors, repetitive domains, transcription factors, cyclins, and so on and so forth. So I'm not going to go into detail in this part. This is more like an exploratory analysis that is the base for future research. An interesting study that we were able to compare between the 4 genotypes of spruce was the phylogenetic tree that we built. So we used the 4 genotypes of spruce plus Picea abies, which is the Norway spruce, and the 2 outgroups of pine in order to see how they relate to each other. And we used several strategies to do so. We used proteins from the genome annotation, the mitochondrial DNA. So you know that, for example, in human, you have mitochondrial DNA that comes from the maternal part, and this is the same for the plants, for the conifers in this case. But plants also have the chloroplast, or more generally, the organellar DNA. And in the case of conifers, the organellar comes from the paternal. So we were able to study the evolutionary history using different strategies and to see how all these species connect to each other. And the first interesting thing that we discovered is that the interior spruce, which is the hybrid that we have in British Columbia, it's actually closest to the white spruce and very also close to the Engelmann spruce. So Sitka spruce, it's a little bit more distant. Very interestingly, so I just want to highlight this. In A and B, so in the proteins and the mitochondrial DNA, you can see that the topology of the tree has Sitka spruce close together to the Canadian spruces, and the Norway spruce, which comes from Norway, Eurasia, it's actually more distant. Instead, in the chloroplast, the topology it's inverted. So you have the Norway spruce that it's closer to the other spruces, while the Sitka spruce it's more distant. So this is probably a story of ancient introgression, where probably there is an ancient traveling of the pollen and the hybridization of the Norway spruce given by the study of the organellar DNA. That means that there is an ancient connection between these spruces. And it's not everything points to the fact that even if they are close in the Canadian continent, they should be also related. Having Norway spruce means that these species were even more connected in the past than we thought. So this study, it's unique because we studied everything at the genomics level. But like there definitely more to know and more to study in this type of analysis. If you want to read more, this is all like together from a thesis and in this publication from this year. So definitely this is a very big effort between several institutes and several collaborations. They are all part of the Spruce Up Consortium, which is a very big collaboration and funding given by Genome Canada, Genome British Columbia, Genome Quebec, and also Genome Prairies, which I forgot to put here, but it's definitely something very, it lasted several years and it was a big effort between the several institutes. And also here I listed some of them, but like definitely there are more parts in the Spruce Up, which I'm not showing. So if you guys want to share and check the publication, there is definitely more to read about these conifers, and it's a very massive work and it's a very big collaboration. So this was the part one of my story, and I just wanted to go in something a little bit related to the conifers, which is basically the biotic stress that some of the conifers that they present, some of a story of another genome that actually it's the spruce weevil, that it's also a pest of the conifers that I just presented. So the spruce weevil, it's a very tiny bug. It's part of the coleopterans and curculionid family. And it's basically, as you can see from this map here, it's very widespread across Canada. And it infests, its range of infestation, it's overlapping with white, Engelmann, and Sitka spruce. What is important to say is that actually this pest, it's able to attack Sitka spruce, and in particular this strain here, Q9 and 3, but not other strains like the Sitka spruce of H898. So some of the trees that we study are actually more subjected to the weevil, while other species or strains are not subjected. So we are trying to understand also what makes the susceptibility of the spruce and why the weevil it's able to attack the trees. This little bug attacks the upper part of the tree, and so it lays the eggs, as you can see in this, like the upper shoots of the very juvenile trees. And this is how it puts the eggs inside the tree with these small holes. And the larvae are feeding on the tissues, and the tree is not able to grow properly, and it's stunted and it's really crippled, so it's not able to reach a proper development as for many trees. And this makes actually a huge impact when you see like forestry or trees that need to be harvested. So this little bug, it's actually one of the major causes of disruption of the forest, especially on the West Coast. For this project, we assembled the genome of a single individual that has been harvested from a population in the northern part of BC. So this is the larvae of the spruce weevil that I'm showing here. Again, DNA extraction and sequencing with 10x genomic short reads, genome assembly, and again, genome annotation. And part of the paper that we also work was a comparative analysis to other species, related species that has been published as reference genomes that we want to know better their evolutionary history. So the first very interesting thing that we discovered in the genome of the spruce weevil was the fact that the genome size is actually very big. So it reaches 2 gigabase pairs, which is about 10 times of other species like the mountain pine beetle that I'm showing here in the panel B, that it's about 0.2 gigabase pair. So there is a very large difference in the genome size. And again, looks like there is a certain gene family of repeats in the genome that it's quite expanded. So I evaluate about 50% of the genome to be covered by repeats. And so plotting the distribution of repeats, so I built a consensus of the repeats that I annotated in the genome, and I was looking at mutations of these gene families in the genome of weevil. And the Kimura plot that I'm showing here shows on the right more ancient repeats expansion, and close to the 0 point of the plot, it's basically more recent. So whenever you see a peak, it's called the burst of repeats, and the dips are extinction. So there has been a very recent genome duplication, which probably also correlates with the expanded genome size of the weevil. And respect to the other species, it has a different profile, especially of the DNA type of repeats and also the LTR type of repeats. So it's interesting to see that we sequence a genome that hasn't been studied before, and it showed a very unusual evolutionary history. And this has been also confirmed by experimental data. So we have flow cytometry that showed its expanded genome size, and it's definitely something that it's worth to study better. As part of the study, we compared the spruce weevil, that it's of this family of the Molitini, also to the bark beetles, and it shows that actually its genome is quite different based on single copy genes that we annotated. And what it's really stunning here is that using the information that we have about the primary plant host and the host interaction, it's possible to see that even if our spruce weevil actually attacks the gymnosperm and the spruce, it doesn't group with species that attack the gymnosperm. So the host it's not a factor that influence the evolution at the molecular level. It's possible to see that host interaction and the type of host that attacks, it's actually independent of the evolution. And for me, it was very, very interesting to observe for the species that we analyzed here. This is part, again, of the bigger collaboration. It's part, again, of the Spruce Up Consortium, but also of the Canada CanSeq 150 initiative that aims to sequence 150 species and to release the reference genomes of species that are important for the Canadian economy. So feel free to check the paper. And we have also, I don't know if I have much time to discuss today, but we found an endosymbiont of the spruce weevil, which we identified not as a contamination, but as a, let's say, a symbiont. So the Wolbachia species that we assemble, it's actually very interesting to study. And we released also the reference genome of this bacteria, which is something that people are quite interested to study in the interaction of host and bacteria. So just to summing up, definitely we have been releasing annotation resources and genome assemblies that we shared with the community. You can find all the reference genomes on NCBI and also shared through the BCGSE site. And having all these genomes, we were able to do a whole genome, like very large scale comparative analysis, which people often do with one gene at a time. But having the reference genome make it a little bit more high throughput and looking at different elements of the genome. And furthermore, we have phylogenomic and also genome evolution analysis. And with this, I just want to say that this is definitely the first stage of the study of genomes for forestry. So we have the reference genomes for the spruce weevil and also for the 4 genotypes of spruce. So having reference genomes, it's going to be very important to study the population of these spruces. So as I showed, some of them are really widespread and they have very different localization, even if they belong to the same species. And studying more individuals from these conifers and also looking at the diversity of the spruce weevil and how widespread it is, it's definitely something for the future.
Oh yeah, okay, perfect. Because I thought that it was blocking the screen, but that's okay. Yeah, go ahead.
Yeah, I'm actually I'm finished. So I just wanted to thank all the people working at the consortium, um, and I think like having without the supervision of the people would I would definitely not was not able to do it myself. And just wanted to thank my supervisor, Dr. Inanch Birol, and my supervisory committee that helped me with framing my thesis, and also for the support for research, and also the people that definitely contributed to this project as a direct interaction and collaboration with me. So with with this, I just want to thank you for listening. And yeah, if you have guys any questions, I'm really happy to answer them. I hope I didn't go too much into detail, but I'm happy to show other slides with more details if you have specific questions. Thank you.
Thank you so much, Kristina, for your great presentation and very interesting. As I have a master degree in plants, I'm like, oh wow, this is interesting. This is interesting. I know how it's very challenging to work on polyploid species and to filter more about the repetition.
It's actually it's not a polyploid, it's diploid. It has only 2 copies, but it's very large. So it's very unusual for plants with not being polyploid to grow so big. But conifers and gymnosperms, some of them they really reach a very big, very large limit of genome size.
Perfect. I thought that it was polyploid because you said that there is seven copy more than the human.
It's like
And then
7 times more, but like it's 2 copies. Yeah.
oh, okay. So yeah, and then you said that there is haploid and diploid tissue. So I was a bit confused, but that's okay. That's great. Was one of my question. But I'll. We have more question now. Ahmed has a question. He said, "How did you assess your genome annotation without completeness? In there any significance test scores for the quality, for instance? How would you calculate it and forward your phylogenetics analysis? Which program did you use? Any accuracy, similarity scores?"
Yeah. So as far as I'm concerned, like the best method known in the community is to use what has it's expected to be annotated. So the BUSCO score And also there is this other score here for completeness, which is part of another package that looks for PFAM domains. So these are the two scores. We don't have any statistical tests, and like I'm always pushing the community to have a little bit better testing for that. I know that this is a weak point, but we are looking for things that we know, and there is not really a statistical test for that. So like 2 tools, and this is all I did for the completeness. As part of my phylogeny, so here I used single copy genes, and I built basically the phylogenetic tree for each of the proteins, and I use Astral, which is like a tree-based type of phylogeny from using genes, and then you have like multi-species coalescent method to combine them in the species tree. And there are other methods. For example, here in B and C, I used a k-mers-based approach. So I look for the overlap of the k-mers. And just to validate if B and C was kind of a method that we wanted to use further, I also used standard method, which is the D method, which is the maximum likelihood with RexML, which is also another software that looks for like it's based on alignment of multiple proteins, and like let's say like A and D are the most known methods for phylogeny, and k-mers method, it's something you do fast, but it looks like it confirms the other two methods that we were expecting.
I don't know, Ahmed, if you have a comment or another question. Ahmed?
Thank you. It's,
Okay, perfect. Sorry, I thought that you were not here. I think Nicola, he wanted to speak. Let him ask his question. Okay, perfect. No worry. Okay, so I have a question too. I just said something about this. So you use RexML at the end, or you use all the software?
So at the end, I used Astral, which
Astral,
built the species tree, and I use several methods. So when you have like what I learned from phylogeny is that you try to use several methods, and if the story doesn't match, it means that probably there is more to know about the species relationship, and like I always recommend to use multiple methods for phylogeny.
Okay, perfect. And do you have a reference, or it was the first study? As you said that it's the first study of genomics, comparative genomics. How did you compare?
Well, we compared. So basically, what you do in comparative genomics, you have species that you want to compare to others. So there was another genome that has been published in 2013, which is the Norway spruce, and we compared to the pines, which are the outgroup in this case.
Okay.
So yeah, we do have like another spruce that is sequenced, and other conifers are known.
Perfect. I can imagine that you use Compute Canada to calculate everything, right?
Well, I think like for the phylogeny, not much. You can actually get a good, also not super powerful computer. You can do it. Depends also on how many. I use a jackknife to basically find the supporting evidence here, the numbers on the notes. But like we have our own servers at the Genome Science Center, so we have very powerful machines. And especially for the genome assembly, that was a very big challenge because these genomes are, so the amount of data to input to generate the first brown graft for the assembly, that was impressive, and the memory you need, it's high. So I don't think Genome Canada, it was probably the best way. So we have proper machines at the institute to do so.
Nice, that's great. Because it takes very long time to do one small analysis.
Yes.
I had some struggle on that too.
It's horrible, yeah. We
Um,
like, I even know, like, when you run a software for 1 month, and it's probably it crashes at some point, and then you say, "Oh my gosh, I wasted 1 month waiting for this
I
software."
know.
That's horrible. So
I figure.
yeah.
Um, I have a question about the heat shock proteins. So you said that you use heat shock proteins for adaptivity, right? And why did you use only two groups, HSP70 and 20?
So these are I did a very in-depth literature study, what has been found already in local adaptation in conifers. And I basically, this is a selection of the genes that I found that has been flagged already in previous studies. So there is no functional explanation, but these two proteins, they pop up quite often in the analysis. So this is like a preliminary study. It's not like a proper functional study that I know the mechanism or how it
Okay, I
works.
see. I
Yeah,
see. So you don't know the link of the adaptivity on that. Okay.
not really.
And you said generally the heat shock proteins they use for when the plants stress, it's overexpressed the HSP70. So usually they use it for evolution. There's a lot of study for
Interesting.
evolution in HSP heat shock proteins. Um, for the significant or no significant, so this is only overall, but is it some explanation behind what's significant, non-significant? What
Yeah.
are
So this is actually like a slide of very long analysis I did. So this is the gene family's expansion, which is basically, I don't know if you guys know the cafe analysis, like gene expansion and contraction analysis based on phylogeny. So it uses a phylogeny tree to simulate how you expect your gene families to expand or contract. And like if what you observe is like different than the expected, it gets a significant score. And like the other analysis, it's a positive selection. So I only take single copy genes, and I use the CodeML. So that's another software for finding positive selection. Uh, Basically, it's based on the synonymous non-synonymous ratio of SNPs on protein level in this case. So this is like a very broad summary of what I spent a few years to do, and to be sure that I have all the controls, and like the comparison was kind of fair between the species. So it's based on two different software and approaches.
nice? Um, the last question: what's the most challenging thing that you did when you assembled the genome of the species?
So what's the most challenging analysis you mean? Like
The step, more challenging step.
yeah, I would say, so the thing is, um, I think it for me what was the, let's say, the comparative analysis between the genomes. So like you basically run a pipeline. Okay, you run software to assemble the genome, you run a pipeline to make the annotation, but when you're comparing the different genomes together, uh, you need to be like properly sure that there is no bias that can influence the properties of the genomes when you compare it to the others, because like of course there are misassemblies or like misannotations, and the important thing was to make a proper filtering and to be sure that you fairly compare like step by step the genes in a way to get meaningful results. So it's definitely putting everything at the same level, because like, oh, these guys here, oh, these 4 genomes were slightly different, even if they were made with the same approach. Slightly different, of course, in the sequencing and the assembly steps, but I tried to make to put them at the same level for the comparative analysis. And this is very challenging when you do comparative analysis, when you have, for example, genomes that you didn't annotate or you didn't perform, but you need to compare them to your genomes. How can you do the most fair comparison in order to get meaningful results? That's challenging.
I totally agree. Another question. You use for the sequencing, you use only the RNA-seq and not the whole genome sequencing, right?
Sorry?
For the sequencing, you only did RNA-seq type of sequencing and not the whole genome sequencing.
We did, so for the sequencing, we did whole genome sequencing. And like the RNA sequencing was mostly used for the annotation stages.
I
So
see.
we have the entire genome, and actually, we have more than 100 times coverage of these genomes. And it's very challenging to have all this data to do the assembly.
Perfect. And for genome analysis, whole genome sequencing analysis, how much time it takes you to do one genome, whole one genome?
Like for the annotation or for the assembly, you mean? Like genome
For
assembly?
for genome assembly.
I would say it varies. So I would like first to thank my collaborators for that, because they took care of this huge project that was even before me for the assembly. So it was building the tools for these massive genomes, and also like making it possible to run on the big genomes. But I would say like starting a new genome assembly for this very like 20 gigs huge genomes, it takes several months. And like once you have one, you want to improve it, and then probably you need to go back several stages, do another for example, scaffolding stage, polishing stage. So it's more testing, and then finding the right like what makes the best contiguity and completeness. So it's, I would say, having the proper genome, it's from 6 months to 1 year, like having a very like exploring all the possibilities to have a high quality, like the highest quality you can achieve with this data. So it's not something to underestimate when you start assembling genomes of species with large genome sizes.
Yeah, for sure. It's very heavy as a data. I
Yeah.
do human genome, and it was very challenging to analyze
Mm.
whole genome sequencing. So that's why I was asking. But yeah, 7 months. It's very long time.
Yeah,
You cannot do some
definitely.
mistake on it.
Well, you always try different approaches. Like if I do this stage of scaffolding with 10x before the stage of scaffolding with probably long reads, is it going to improve? So you need to try it, and you need to give time to yourself to do it. So it
Hmm.
takes the time it takes.
Yeah, I totally agree. Perfect. Perfect. I don't know if you have question. 3, 2, 1. Okay, perfect. So thank you so much for maybe. I mean, he has a question. No, no. Perfect. So thank you so much for your presentation, Kristina, and thank you all for being here and attend to the monthly meetup, MonBUG. So we have another meetup on December, which will be the last for the year. It will be a good presentation to high quality as today. So thank you so much, and see you next month. And yeah, just to let you know that the next week there is a 5 set at UCAM. and it will be on Wednesday. Please check on the meetup.com MonBUG to see to see more of the detail about the 5 assets. So yeah, have a nice evening.
See you. Thank you.